Let's get started here. First, break the ice. I want to show this clip. That's just to give you an idea for those of you who haven't seen a bioprinter. So this is the newest bioprinter organ regenerator 46 from Rocket Healthcare, about which I'll talk later. And for the people who are totally new to bioprinting is a perfect combination of computer science, biology, instrumentation, engineering, and material science. It brings the benefits of personalization, autologous solutions, and fine details to human health applications. So it is bioprinting, 3D bioprinting is an additive manufacturing process, just like regular 3D printing. In fact, it is 3D printing, except that it involves cells. Um, so it involves cells, biopolymers, growth factors, all combined to create tissue-like structures that imitate native human tissues. Because these printed living tissues are often further incubated to have them proliferate, mature, and change over time, there is a dimension of time here that makes it appropriate to call this as a 4D bioprinting rather than 3D bioprinting, which we all hear often. So you will hear me use these words 3D bioprinting or 4D bioprinting mixedly, but you get the idea that there is an important temporal component because just printing a structure doesn't make it functional. So just keep that in mind, but otherwise the terminology should not be a big deal to understand the concepts and the execution. And to keep it less distracting for myself and everyone, I'm going to turn off my video if possible so that I can focus on the presentation here. And I would encourage you all to do that as well. So, and we can rejoin, but it's up to you. 1 second, please. Okay, all right. All right, so first I want us to take a look around and see what's happening in the news over the last 2 years. So there are 5 notable events and megatrends that really warrant our closer attention to the growing relevance and potential of bioprinting as a revolutionary technology for adoption into biomedical research and in the treatment of diseases. And the first one is in September 2018, we saw the featured news of a boy who was living for 14 years with a new urinary bladder made from his own cells, seeded with 3D inkjet machines into decellularized pig bladder as a scaffold. Now, this work and similar work by many others, it clearly highlights the promise and the potential for bioprinting of human tissues to transform medicine with implications for organ transplants, cancer treatment, and many other possibilities, such as develop new antibiotics and drugs efficiently. The other thing is in July 2019, the American Medical Association approved reimbursement codes for 3D printed surgical models and tools. Now, this is a temporary code, but nonetheless, it's significant because these are making the foundation for a clinical registry of patient 3D scan data and relevant 3D modeling, which will serve as a learning resource and tutorial and provide the much needed impetus for active implementation of 3D printing in biomedicine. So that's important. Now, in in September of 2019, later that year, United States Environmental Protection Agency announced that it will eliminate all mammal testing by 2035 and becoming the first federal agency to make such a strong statement and commitment to the transition to a humane research while emphasizing the urgency to find alternate solutions to the use of animals in research. Now, later that year, in October, Rocket Healthcare and a diabetic foot ulcer specialty clinic in India, they put the bioprinter in the operating room to create a streamlined process of skin regeneration for application in diabetic foot ulcer treatment. So this new and alternate method for chronic wound healing promotes skin reconstruction using bioinks made from patients' own adipose tissue, printed into small size-specific patches and applied directly onto the wounds. Now, these dermal patches integrate with the skin, and through early onset healing, Rocket's mission is to decrease the rate of amputation for patients with the diabetic foot ulcer. Now, this is especially critical because the 5-year survival rate of patients who have undergone amputations is way lower than the 5-year survival rate of many cancer patients. Now, last but not the least, in the most recent months of 2020, COVID-19 has changed everything about the way we live, and most notably, we have seen that this pandemic disrupted the traditional centralized value chains in manufacturing and healthcare. For example, when the hospitals got cut off from the supplies of masks and ventilators because factories didn't function because of the pandemic lockdown, the 3D printing and additive manufacturing processes came to the rescue to manufacture these supplies on demand, on site. Now, why am I saying all this? When we put all this together, what's the take-home message from these global events and megatrends? It points to the fact that 3D printing, additive manufacturing, and as an extension of that, bioprinting can react directly to centralized demands for life sciences research and can serve as an effective medical strategy. Clearly, there is a growing value for on-demand biomanufacturing, and bioprinting is quite relevant to the real-world needs and megatrends. Now, it is for this irrefutable connection to the real world that I'm here today to talk about the 3D bioprinting technology. For reasons that I just discussed, it may be worth paying attention to how this technology is maturing to impact our research and healthcare workflows today. Now, we have been hearing about bioprinting, and it's getting a lot of attention in the mainstream research, but only recently. But that doesn't mean that the origins of this technology are new. In fact, the origins of this technology go back nearly 2 decades. So for those of us who are still debating if this is a very new technology that on which one can wait for several more years before you consider adoption into your workflow, the news is, no, it's not a nice thing to do because the technology is not totally in its infancy. In fact, the number of research publications, as you see here in this graph, has been steadily going up. And it's been growing exponentially since 2000. And today there are thousands of publications as a testament to the benefits and the applications of the bioprinting technology across multiple disciplines, ranging from tissue engineering, biomaterial development, regenerative medicine, and so on. Now, it's important for us to keep in mind that bioprinting is a platform biomanufacturing technology with not a limited scope in terms of application to any one narrow field, or one shouldn't assume that it is exclusively in the domain of only regenerative medicine and it can be used anywhere else. No, it is, as I mentioned, a platform technology. So in this slide, you will see that there is a list of publications that are hyperlinked, and you will see that there has already been a lot of evidence about ideal choice and combination of cells and biomaterials for really tissue-specific applications using bioprinting for various tissues: cardiovascular, musculoskeletal, neuronal tissue, hepatic tissue, and so on and so forth. And then a huge inventory of bioink polymers and what type of concentrations and applications. So all of this knowledge means that it is a platform technology that you can take and apply for your own area of expertise or area of research focus. Now that's important to keep in mind as you consider adopting this. Now, one other major challenge in medicine is not having a sufficient number of tissues and organs to replace the failing and aging ones in patients. Modern tissue engineering aims to indirectly replace damaged tissue with man-made scaffolds that support cell growth. Bioprinting is at the frontier of this field because it is the only platform technology that can create these tissues and organs with applications in all these different types of diseases that come with aging. There is actually a lot of evidence about the ideal choice, again, of combination of cells, biomaterials that one would need to for a specific tissue application involving bioprinting. So all of that information is out there and it's growing fast, and all of that needs to be taken advantage of. All right, so time to address the basic question. What are the key components of bioprinting, be it 3D or 4D? The bioprinting technology consists of largely three components. One, biomaterials. Two, bioprinter, which represents the hardware component. And then we've got the design element, which is predominantly the software. First, let's talk about the biomaterials. Unlike the traditional 3D printer, which uses hard plastics or metal or any synthetic material to create non-living structures, as I mentioned before, the bioprinter uses materials that are biocompatible, biodegradable, and they are derived from either natural sources or synthetic sources. Now, some of the common examples of naturally derived biomaterials include collagen, gelatin, elastin, chitin, coming from extracellular matrix of human or animal cells. And it also includes materials like alginate, sericin, or cellulose, coming from seaweed, insects, plants, respectively. Natural hydroxyapatite is also used, for example, coming from the mammalian bones. And then in terms of the synthetic biomaterials, these are basically biodegradable polymers with both strength and elasticity to mimic human tissues. And some of these have been put to good use in FDA-approved medical devices. And finally, you also have composites, which often combine natural and synthetic materials to add or enhance certain mechanical or chemical properties. Now, these biomaterials can be soft, low viscosity hydrogels or hard biodegradable plastics. And when hydrogels are combined with cells for printing, they are called bioinks. Bioinks basically serve as the extracellular matrix when you're trying to print cells outside the body. And bioinks, they protect the cells from the shear stress of the printing process, and they provide them with nutrition and hydration, and they give the much needed overall physical chemical support to maintain the structural integrity of the printed tissues. So That brings us to an important point that when selecting a material for bioinks, there are several key criteria to keep in mind, but they can basically be summarized into two broad categories. One is that the bioinks should be able to extrude out of the nozzle tips and stack layer by layer structurally with or without any post-printing processing, about which I'll talk later. Now, the bioinks should also be biocompatible and should uphold very high cell viability. And these are some elaborations of the same point of the different properties. And why I focus again so much on the biomaterials is because, as I said, of the three pillars, which is the biomaterials, the hardware, and the software, the biomaterials are often the rate limiting step for any particular new application. All right, let's move on to the hardware. There are various methods of bioprinting. There are various methods of bioprinting today, as you can see here in this diagram. They include methods like inkjet-based, laser-assisted, stereolithography methods, and so on. But of all these, the one highlighted here, the extrusion-based method is the commonest one. And that is also the method that we will focus a lot here today. One key component of any bioprinter hardware is the print head itself. It should be able to print both hard and soft materials. And there are two ways that the print heads can print. One is print in a scaffold-laden way, where you print a hard matrix, and the other is a scaffold-free manner in which there is no hard matrix. Another key component is the chamber's ability, the bioprinting chamber's ability to mimic a biosafety cabinet, because after all, when you're dealing with living cells, it's important to ensure a biosafety cabinet environment. To optimize the cell viability and functionality under the stress of printing, the chamber should have a UV disinfection and a HEPA filter and some type of bioreactor capability to provide the optimal environments for cells, especially when it comes to temperature and humidity control. And lastly, the third component of bioprinting is the design, which means the 3D models of the objects that we want to print. Objects meaning, in this case, tissues or organs. 3D model files, which are known as STL files, they can be obtained either by manual creation using a design software, or they can be derived from a CAD or MRI scan of a physical structure like a human body or a body part or an organ. Now, these STL files then get processed into a slicing, with a slicing software, into a standard machine language file, which describes how a 3D printer should print the object or the structure in a given three-dimensional space. It includes information on the X, Y, Z position and the coordinates, printing speeds, infill patterns, and numerous other parameters that are important to recreate the structure from the digital file to reality. The example images here on the bottom kind of help you visualize the different, the rendering of these in the different files. All right, so now that we understand the important components of bioprinting, we can put together the bioprinting process logically. The process principally involves 5 key steps. To summarize briefly: preparation, which involves designing the experiment with a clear identification of the problem to address. It includes selection of the right materials, cell types, and what models that the printer will use. And printing, of course, is the actual process of printing, and 3D models are sliced into appropriate printing instructions. The bio ink is placed in a printer cartridge, and the deposition of the material takes place based on the digital model. And then we've got the post-printing processing. You take the printed structure and do a mechanical or a chemical stimulation as needed, and it can vary depending on what material has been used, so as to create a stable and functional structure for the biological material. And post-printing can involve cross-linking, thermal methods, chemical methods, or differentiation, incubation, and the air liquid, whole bunch of things. Those are just the fine details which we won't worry about. Uh, Evaluation is the step in which you make sure that the printed tissue is assessed for structural, morphological, and functional characterization in comparison to a living tissue standard. And lastly, you do your experiments with the printed tissue, or if it's a therapeutic application, then you do the therapeutic application. So it involves use of the bioprinted products for various purposes, ranging from disease modeling to in vitro safety, efficacy testing, or in vivo tissue transplantation. Now, to the same workflow is shown here as an example of cardiac patch engineering. What would you do? What are the first steps? Say you take the ischemic heart disease scenario, and then there is a need for cardiac regeneration. So you prepare endothelial cells and so on and so forth. So in the interest of time, it's very intuitive. So in the interest of time, I will skip this. You will get a recording presentation, which you can refer to later on. Now, of course, cardiac patch engineering, which I just mentioned, is just one of the many, many possible downstream applications with bioprinting. So bioprinting has been, as I mentioned before, in various areas across tissue engineering, material development, regenerative medicine, veterinary medicine, toxicology, pharmaceutical sciences, and so on. And the list is growing. So for simplicity, one can summarize the use of bioprinted products into three key areas. First, bioprinting for toxicology research and drug screening. So this is mainly to be used in the in vitro in generation of in vitro efficacy test models for toxicological testing and screening of novel products, whether it is a drug, a cosmetic, or food. For example, in an ideal study to evaluate the toxicity of a chemical or substance to humans, one would require an extremely large number of human subjects to represent the diversity of humans. Now, we all know this is unrealistic and unethical. So the use of human relevant tissue models that are derived by bioprinting will offer an alternative and will be a good bridge between the human clinical trial and the animal studies with the standard human tissue fabrication process that's high throughput and pretty standardized. So that is one area of application, strong application for bioprinting. Now, moving on to the second big category of application would be to create in vitro disease models to study the tissue physiology as well as disease. Now, traditional science, we all know, has relied a lot on the 2D cell culture and 3D spheroids, about which Dr. Panos just mentioned. But decades of research has shown that the human tissues are hyper-complex and they are heterogeneous and structurally, mechanically, biochemically. They are heterogeneous and complex. So considering that, for example, let's say cancer or tumor is a good example of such complexity. And bioprinting technique can help in this, in the recreation of in vitro study models for disease like cancer, because it has the ability to fabricate and visually map tissues in 3D, enabling us to better and accurately understand the disease and the consequences and also help us to eventually develop accurate therapies. Now, third is the kind of the holy grail of all of this, which is to be able to uh, use bioprinting approaches in, in generating in vivo models for transplantation and regeneration. Now, one of the most common challenges in medicine, of course, is not having the sufficient number of compatible tissues and organs to replace in the events of trauma and organ failures. Flat tissues like skin, bone, or tubular structures like blood vessels and urethras, they may be relatively simple, but hollow non-tubular organs like bladders and pumping organs like heart, lungs, they are very difficult to replicate, and not to mention the complexity with neuronal tissues. So for such highly complex tissues to be created, the bioprinting technology will provide a high degree of precision that surpasses human hands or any other existing technique. Now, if these could be made with patients' own cells, then the challenge of immune rejection and donor shortage with organ transplantation can also be resolved once and for all. So along these lines, so this one here is to give you an impression of what applications are possible. Now, what you're seeing here is a bioprinted cardiac patch that behaves very much like a real cardiac tissue. And you can see the pumping action. Now, as an extension of this same dream, let me pause this. So as an extension of this same dream of creating viable tissue for transplantation, one can imagine that in future, we can perfect the technology to the point where we could incorporate nanotechnology advances and smart materials can be incorporated into the design of these organs and tissues. And that would lead to more creation of smart organs that would have extended durability that would be unimaginable for any naturally occurring tissues or organs. So there are a lot of interesting possibilities that exist with the bioprinting technology as it matures. All right. So in the context of all of these we have discussed so far about bioprinting, I want to again mention that Rokit Healthcare is an emerging key player in this space, in bioprinting and organ regeneration space, through its innovative products and services. And apart from that, its robust in-house R&D programs aimed at creating breakthroughs in science and healthcare. Rokit is dedicated to providing these innovative offerings to well over 25 countries around the world, with more than, I guess, 250 user institutions and partners, including many here in the US. So as a leader in this space, Rokit introduced the state-of-the-art bioprinters, Dr. InVivo 4D, as the world's first clean chamber bioprinter with HEPA and UV lamps and hybrid print heads for both soft tissue printing and hard tissue fabrication. And as I mentioned and showed in the early part of my talk, they also recently launched the Dr. InVivo 4D6, which is the world's first bioprinter / organ regeneration platform to come fully integrated with cell incubators and plasma sterilizers, along with 6 different print heads and so on. I'll talk more about this later towards the end of the talk. Now, let us look more at the important part of our talk today, which is the applications and the publications that have come out using bioprinting for various different areas. So the slides that I'll be showing in the following are going to be mostly summaries of the publications involving or using Rokit's Dr. InVivo 4D. And so it is basically a lot of data from Dr. InVivo 4D users. Plus, here and there, you will see some data from users that haven't used InVivo, but a different platform, to impress upon the potential of bioprinting technology. Now, I'm not going to go into the details of all this. It's practically impossible. to cover all of that in a short talk, but I'll touch upon to give you an idea or to impress upon you the diversity of applications without going too much into detail. But if you have any questions, reserve that to the end and then let me know, and we will guide you to the publication, and you can look at it, or if I know the answer, I'll let you know right away. All right, so let's start with the applications in the nervous system, which I believe is of most interest to you all. The peripheral nervous system and the central nervous system are some of the hardest tissues to regenerate and repair. There is no denying that. Now, in case of the peripheral nervous system and central nervous injury, traditional neural tissue replacement strategies, including the autogenous nerve grafts or nerve graft substitutes made with autogenous vessel, muscle, or decellularized autografts. Although the autogenous nerve graft is still the go-to strategy, there are significant limitations in creating another wound to repair the original defect. So because it results in a significant morbidity of the new donor site. Now, I'm trying to go over some of the current challenges or problems in this space. Also, one other challenge is the fact that there is the mismatch between the size and length of the donor and the recipient nerves. And that can be a problem leading to poor regeneration capacity. For these reasons, the use of artificial nerve guidance conduits made with biocompatible materials has been on the rise lately. However, with traditional tissue engineering techniques such as electrospinning and scaffolding, there is very little control over the complexity of architecture, whether it is in terms of material composition or the structural design. With other tissues as well, this has long been the problem. Now, to overcome these limitations, 3D bioprinting has been rising to the table to offer an ability to make free-form designs with various types of cells and biomaterials. Now, despite the fact that its application in peripheral nervous system or central nervous system regeneration is still very new, it has, by it I mean 3D bioprinting, has made progress over the years. Now, this figure here, as you see, will summarize where each of these applications are lying today in terms of the developmental stages that they are in towards a clinical or commercial use. For example, you see on the left, you see scaffolds for central nervous system repair are still at a very early stage in development, as are the 3D printed in vitro brain models. Now, towards the right, you see 3D printed scaffolds for peripheral nerve repair that are slightly further in development. And similarly, we also see that the in vitro models for peripheral nerves with relative simplicity are also on the far right end. Now, in modeling the peripheral nervous system, a main challenge is the incorporation of multiple cell types into specific patterns. Now, in this context, in this particular context, 3D bioprinting offers the most advantage by being able to position cells in multiple compartments or micro grooves and aligning them the way they should for any optimal axonal growth. For example, as shown in this figure, and this is a figure taken out of a publication that came in Lab on Chip by Johnson et al., and I can provide those references later. What they have done is they've created an in vitro device for studying the peripheral nervous system that resembles a mini nervous system on a chip. This 3D printed device contained 3 chambers, 1 for peripheral nervous cells, 2 for Schwann cells, and 3, as you can see here, for terminal cells. Now, it's sort of like the way the peripheral nervous system would be structured in its simplicity, of course. In addition, the device contains parallel microchannels from silicon and a sealant layer right over them. This way, the Schwann cells and the terminal cells interact with the neurons and each other solely via the axonal network guided by the silicon microchannels. Now, if it's all confusing, it's simply to keep in mind that you can design and fabricate the way you want to control the interactions. And that precision and ability to come up with these combinations is possible with bioprinting. In many other techniques, most other techniques just can't even come close. In the article, the authors not only demonstrate the basic cell viability and structural mimicry of the tissue, they also demonstrate the ability to track a viral infection as it spread from one cell type to another within the multiple chambers of the simple model peripheral nervous system device. Now here, what you see on the other hand is modeling the central nervous system in vitro. Now this has far-reaching implications for studying brain and spinal cord function that would otherwise be difficult to examine in vivo. Already over a decade ago, several research studies have shown that printing human neuronal stem cells into 3D structures and then kind of differentiating them into mature neural cells, it leads to a higher expression of differentiated neural cell markers than the same cells cultured in 2D. So the same was seen in the induced pluripotent stem cells in this study, following the same process. Bioprinting in 3D also permitted the creation of complex structures that recapitulate important aspects of brain structure. For example, the human cortex contains a distinct 6-layered structure. Recreating this structure in a controlled manner is very difficult to do manually or by any other technique, but it can be achieved efficiently through 3D bioprinting, as you can see in this figure from the publication. And I think this publication is from Lonzano Group. Yeah, and came out in Biomaterials in 2015. So basically, the group used a bioink that consisted of a novel peptide modified biopolymer called Gellan gum, that is written here as RGDGG. This was combined with primary cortical neurons. The Gellan gum is a polysaccharide obtained from microorganism fermentation and was first introduced as an alternative to agar and gelatin in the food industry. Now, in tissue engineering, Gellan gum has shown many benefits over the other hydrogels, such as low production cost, biocompatibility, and achieving similar levels of mechanical strength as gelatin or agar at a lower concentration. Now, in this study, the research group well demonstrated that they could create 6-layer brain cortex structure, whether in vertical or planar orientations. Although still at a very early stage of development, this example highlights the potential for 3D bioprinting to create a complex 3D in vitro model of the brain. I mean, there are going to be a lot of opportunity and scope for improvement to the next level, but there is the strong promise and potential, and that's what I'm trying to impress upon you. All right, now this is a publication, as I mentioned before, from the Dr. In Vivo user, which is Rokit's own bioprinter in Korea at one of their premier institutes, the Korean Institute of Science and Technology. In contrast to the in vitro models we discussed so far, this one showcases the use of bioprinting to create regeneration strategies in vivo for peripheral nerve injury sites using a nerve guidance conduit. In this study, what the group did is they developed a biocompatible elastic nerve guidance conduit And they first made a nanoporous membrane of PLCL material with electrospinning, then rolled it up and designed the interior with neutralized oriented collagen hydrogel patches that were printed in high resolution using Dr. In vivo 4D2 bioprinter. Now, by the way, the PLCL is a very widely used biomaterial in applications such as wound healing, formation of cartilage, and cardiovascular applications because it has excellent biocompatibility and biodegradability, as well as high elasticity and flexibility. Now, these are all some of the things we discussed talking about the right choice of biomaterials a few slides back, compared to the regular PCL or PLA materials. Now, collagen is also a widely used bioink because it makes up a very large percentage of any natural ECM, close to 70–80%. So the bottom line is the group compared the efficacies of PLCL conduits filled with bulk collagen and then 3D printed collagen, as you see here in the panel here of images. And what they found is that in an in vivo study using the sciatic injury model in rats, the 3D printed nerve conduit guidance group exhibited denser and more organized myelinated axons than the group with that was made with bulk collagen hydrogel. The group concludes that the precisely aligned collagen hydrogels enabled by 3D printing patterns of grooves appear to provide a preferable environment for nerve regeneration, providing a guidance path along the longitudinal direction. Now, this was true both for axonal regeneration, remyelination, as well as functional recovery. Now, in conclusion, this is a great example of how bioprinting can help with regenerative tissue engineering by providing an architecturally suitable environment inside the conduit for axonal growth. Given that traditionally the Schwann cells have a poor ability to migrate and align through gaps, the ability to make the paths by designing the interior with bioprinting can probably offer a crucial advantage to effective axon regeneration across the gaps. All right, as we could see from these examples of the in vitro neural tissue models and the nerve systems, bioprinting has three major advantages. In summary, so the 3D bioprinting offers rapid free-form prototyping capabilities, allowing for fast fabrication of living tissue constructs of diverse 3D topologies compared to existing tissue engineering techniques. Also, it has an excellent ability to construct tissue models with a homogeneous cell distribution and uniform spacing. Now, that gives an advantage of repeatability of the experiment, which is very important. Bioprinting also provides exceptional versatility in cell positioning, whether it is vertically in full layers or planar in different cell compartments, it can be done. Now, these are all crucial factors to study a living system with complex biochemical interactions and architectural features, and bioprinting is a good technique that has the potential now, and with more improvements and progress, it can do more towards that cause. All right, now what we discussed about neuronal tissues and the nervous system applications of bioprinting is just tips of the iceberg when it comes to bioprinting applications. Bioprinting is, as I mentioned time and again, is a platform technology able to accommodate various cell types, biomaterials, 3D designs, and purposes. Now, each user with their own creativity and innovation, the applications are endless, literally. And so it varies across multiple areas of application. And so in the next few slides, what I'm going to do is Without going too much into the details, I'd like to give you an overview of what are all the different types of applications that some of our existing in vivo bioprinting users have done and published. Uh, so this is a good example of the first example I have here is developing novel biomaterials with bioprinting. So in this study in Shard, which was made in Hanyang University of South Korea, a group developed a synthetic self-healing hydrogel called OHA GCADH. For now, let's not worry about um, the names. But what they have done is, as you can see here, that is the pink and the white gels here. Now you cut them and then you let them to adhere to one another on their own. So that's a self-healing gel that was made possible. And using the in vivo bioprinter, this biomaterial group was able to innovate this material. And they believe that this self-healing hydrogel system may have potential in tissue engineering applications. All right, now this is an example of adipose tissue engineering using bioprinting. In this study, briefly, the research group from Seoul National University, they sought out to engineer a novel way to regenerate adipose tissues. Conventional medical grade materials such as PCL are often used in breast reconstruction and nose reconstruction, but they do not match any mechanical property of the adipose tissue. So this group developed a novel material using PLCL and mixed it with adipose-derived ECM. And using the bioprinting technology from Rocket, the group created a size-customized scaffold, which when implanted into the mice, it promoted adipose tissue regeneration with greater macrophage infiltration and vascularization. So they think that this would eventually lead to better solutions for burn, trauma, injury, post-mastectomy repair, and so on. Here is an example which I also showed, the beating heart. So application in cardiovascular tissues. At the Fraunhofer Institute in Germany, the in vivo bioprinter was used to develop a patented disc that allows the iPSCs to differentiate into cardiomyocytes, to align and create a beating model. Now check out this video. I'll play this one more time. This is an in vitro heart patch that was printed, and the in vivo printed cardiac patch has been beating for over 6 months in culture. So it is possible to create viable tissue. Now, what we have here is a case where cell suspensions of muscle cells are printed using PCL pillars. So this is an application to demonstrate for muscle model for drug testing. Here is an example. Okay, so this is important, so I'll take a little bit of time to explain this. So lately, more than ever, bioprinting is being explored to create disease models that can better predict drug responses, and cancer is one of the hottest topics in this area. So as we all know, existing in vitro models of cancer are often single-cell spheroids and lack the tumor microenvironments crucial to their intrinsic behaviors and malignancy. Now here in this example, we've got bioprinted layer of lung fibroblasts, the HUVECs, in bioinks of alginate, fibrinogen, and gelatin. Then multicellular tumor spheroids of glioblastoma cells are seeded onto the vascularized tissue, incubated, and resulting in vascularized tumor models. Now, those are being used for drug testing. This is an example of the applications in bone regeneration scaffolds, where in this particular case, a strontium metal ion is incorporated into a hydroxyapatite to create a scaffold that has a higher cell proliferation and osteogenic differentiation and cellular mineralization potential. And as I said, the applications are numerous, and literally it is up to the investigator. Now, in this case, it's an example of developing a QR coded drug delivery device, where this was bioprinted as a oro dispersible film. And they believe that if we keep up with the trend, these type of printed devices can eventually help in replacing anti-cancer injections with oro dispersible films, seeking a more effective and minimally invasive therapy, and also better patient compliance and monitoring of the dosages. Lastly, this is an example of one other interesting application of creating a biosensor. So in this case, they have printed an egg-shaped bioresponsive device that combines microbes, hydrogels, and conductive carbon nanofibers, all in a small space. And this platform offers an improved method for culturing, monitoring microbial cells and their metabolic activities in a confined, organized space. So the potential applications of this type of device is expected to be in studying wastewater treatment, microbial biosynthesis, or metabolism, photosynthesis, so on. So there are numerous applications that I can go on, and I've actually skipped a lot. So I'm going to try and summarize. So one other important thing is we talk about all these applications in research. Yes, that's all great. But the most noteworthy applications eventually are going to be the ones that actually take bioprinting off the bench into the clinic, right? So just to touch upon that, as I mentioned before, the diabetic foot ulcer is a debilitating chronic condition with side effects affecting almost 25% of all diabetic patients. So what Rokit has done here is it has spearheaded a clinical study in India, Korea, USA, and one other country, I guess it's Turkey, where they had almost 100% of the patients in that clinical trial were able to recover from wounds that were basically staying open for months to years. Now, all of them were completely healed within 4 weeks. So this kind of demonstrates the incredible potential for bioprinting in the clinic, especially in skin applications to begin with. And one can only imagine that this can be, with a lot of work, of course, eventually perfected for many other applications. So it's possible to imagine a kind of a special, what we call as the integrated 3D scan print therapy. And in the interest of time, I will quickly scroll through this video. So basically the wound, as you see here, it's not a real leg, of course, it's a model. It's scanned, and then the wound area is digitized, and then the software analyzes where things have to go, and then it recreates the digital file of the wound. And after that, the measurements are incorporated, and the bioprinter prints the scaffold, and patient's own adipose-derived extracellular matrix is incorporated, and then a treatment patch is prepared, and it's applied in a customized fashion. So it is possible to conceive those types of applications. So how do I say this? So I've gone through an array of applications across many disciplines. Now, the key point for you to remember is that this is a platform technology. It is not restricted to any one discipline or cell type or biomaterial. Sky is the limit when it comes to its application, and you can be creative in your own research efforts. So in the last few minutes, I will introduce the bioprinter, because usually we end up doing a demo in person, but thanks to COVID and the pandemic, we have to try and do that virtually. But I will, this is the InVivo 4D. There are two generations of bioprinters from Rokit. This is the first generation, InVivo 4D, and it is the same device that was used in the diabetic foot ulcer projects, and most of the data that I have shown here, in fact, almost all the data. And just last year, the InVivo 4D6 was launched. So it comes with the ability to have different types of print heads, the hardware, to print soft hydrogels, powders, mixtures, powder mixtures like hydroxyapatite, chitosan, hydrogels like collagen, and then also it can extrude filament type material like PCL or PLGA, PLLA, and it can also print using materials that are pellets and powders and pastes like hydroxyapatite that require very high temperature to melt them. Now, as an extension and listening to a lot of feedback from the customers, the InVivo 4D team understood that for the bioprinter to be really usable in the clinic as a true organ regeneration platform, it needs to have much more capabilities that go beyond the domain of research project. And so some of the things that were identified in that analysis were the need for in situ 4D printing, because the human body is a curved structure. So you cannot always think about being able to print or recreate human tissues as just planes. So that led to understanding the in situ 4D printing, curved printing. And then when you're dealing with the applications for the human body, it's important to have perfect sterilization. So the device should be able to sterilize itself as well as the implantable device that it creates. And lastly, the need for a good incubation and humidity and temperature control cannot be overemphasized. So, result, all of these elements were incorporated, and then Rokit came up with the InVivo 4D6, the next generation, that is the true organ regenerator with Z-axis curved printing technology and inbuilt sterilization, and also optional hydrogen peroxide plasma sterilization, and inbuilt incubation systems for maintaining the temperature and carbon dioxide levels during the print processing and post-printing processing. This is a quick overview of the system in action. As you can see, it has a rotary print head of almost 5 different print heads plus one stationary print head on the left. That gives you the ability to deal multiple materials simultaneously to create really complex architecture like the ones that you see here, which are often found in the human body. And you have the ability to keep the cells happy during the process and during the maturation process because of the inbuilt incubator and sterilizer functions of the 4D6. And it's also important to mention that the device is capable of very high throughput and up to 384 well in 384 well format printing, which is especially important for applications such as, say, when you're doing a mini organ drug development project, or if you're involved in a vaccine research or PCR genotyping. So it kind of doubles up as a high throughput liquid handling system as well. So as I mentioned before, all these capabilities provide, and the multiple print heads provide ability to handle diverse materials and hybrid printing technologies and very high temperature ranges, which means you are never going to be limited by what you can do with the equipment, no matter what unpredictable or unknown experimental goals you might come up with in future. Again, so this is not just um, um, a bioprinter for the research lab. It is way more than that. It is a fast track to medical commercialization, because at some point for most researchers, the need to take their innovation to the bedside is going to be important, and the in vivo bioprinters can do just that. This is just to impress upon you um, the user interface, the intuitive user interface, and the ability to control the bioprinter, using the inbuilt user interface, or the tablet devices, or mobile devices, or the computer, whichever one you choose to. All right. So this is really, in conclusion, what's the real essence of bioprinting here? The At the end of the day, the take-home message for you from this talk is that bioprinting brings three breakthrough benefits to research and novel therapy developments. One is automation, faster and precise, physiological relevance of what you print, and the on-demand fabrication. Now, all these three breakthroughs, they result in the ability to experiment with better repeatability and better physiological relevance, the ability to generate biomimetic structures and tissues without having to depend on model systems that are either too expensive or simply not ideal, the ability to create more sustainable research methods, phasing out animal models with alternative human cell-based methods of testing. Now, imagine that the bioprinting facilities could be set up in every region of the world. Then the technology has the potential to completely transform regional biomanufacturing value chains, just as COVID-19 gave us a hint of its needs in the past few months. And you could have facilities across that can be promote a decentralized approach to the manufacturing. In fact, this is not just a dream, this is a reality, because in fact, there are bioprinting centers of excellence rising at national and international levels over the last few years already. As you can see, some examples pointed out here in this slide. And lastly, the key paradigm is the endogenous tissue regeneration. We live in an era today of regenerative medicine. And in this era, bioprinting is not only a revolution that's underway for the way we create and use model systems for answering basic biological questions, it is actually a revolutionary way you can practice medicine. Instead of using the systems that have traditionally relied on treatments of symptoms using small molecules or monotherapies, bioprinting technology is shifting the focus towards harnessing body's own healing potential inherent in its stem cells. Now, this is also the vision and mission of Rocket, to see that come to fruition and into actual adoption in the regular practice of medicine. So the future of medicine is already being realized today by some of these pioneering technologies in various disciplines of science. So I will leave it at that. And I thank you all for sharing your interest and being very patient. I will be happy to take some questions now.